Hello AP Biology students, let's go over the 11th section in Unit 6. And in this section we're going to talk about something called prokaryotic regulation. Remember that prokaryotes are cells that lack a nucleus and they're typically smaller than eukaryotic cells. An example of prokaryotic cells would be like bacteria. When we're talking about genes and how they're expressed into proteins, we need to talk about something called efficiency. Basically, efficiency is using resources in the best way possible and getting a good return on investment. This is probably one of the major themes, in my opinion, of AP Biology. How do we regulate things to make cells more efficient? Prokaryotic cells use something called an operon model. And basically this operon model is a way to regulate gene expression. Now there are four main parts of this operon model. You have your regulator, your promoter, your operator, and your genes. The promoter is a short sequence of DNA where RNA polymerase is going to bind in order to start transcription. The diagrams I'm going to use to help explain these concepts are going to look like a line, but this line is just representing DNA. So you can see this promoter region is just a section of DNA where RNA polymerase is going to bind to. Now the genes that I'm talking about are just sections of DNA. They're going to code for either proteins or enzymes in some metabolic pathway. The regulator gene is going to be found outside of the operon, and what it does, it makes a DNA binding protein called a repressor. A repressor does kind of what it sounds like. It's going to repress transcription. Or in other words, repressors are just proteins. They're going to bind to DNA to stop transcription. How a repressor stops transcription is by binding somewhere called an operator. This is a location, again, on the DNA that's going to interfere with RNA polymerase's ability to transcribe some gene. So you can see here, this operator is going to be found between the promoter and the gene. And what happens is this repressor that we were talking about, the protein, is going to bind to this sequence of DNA. Whenever it binds to this sequence of DNA, it's either going to stop RNA polymerase from accessing the gene, or it's actually going to interfere with RNA polymerase with binding to the promoter. Let's look back here at our original diagram of an operon. First, you have your promoter, which is the location where the RNA polymerase binds to. You have your operator, which the repressor binds to. And you have your genes that are going to be transcribed into RNA that makes some type of protein. Outside of our operon, we have this regulator gene that's going to make a repressor. Now, our repressor is going to bind to the operator, and it's going to stop RNA polymerase from transcribing the genes. The whole goal of an operon is to regulate how much gene expression or how many proteins we are producing. This enables the cell to stop transcription and thus stop translation to make it more efficient. If it doesn't need a protein during some portion of its life, it's going to waste materials if it makes that protein. We have two main types of operon models. We have inducible and repressible operons. Inducible operons are operons that are normally off, but can be activated when the presence of a substrate is there. So again, usually they're off, but sometimes they can be activated. The lac operon model is going to be the operon model that most textbooks give an example for the inducible operon. Before we get into this operon, let's just talk about lactose and lactase. Lactose is a sugar. It's specifically a disaccharide that bacteria will use for energy. Lactase is an enzyme that the bacteria will use to break down this sugar. So again, whenever lactose is present, the bacteria will need to produce lactase. However, if lactose isn't present, they won't need to make lactase. If they make lactase without lactose, it's not going to be efficient because, again, the lactase isn't needed. So whenever there's low lactose or no lactose, the repressor is going to bind to the operator. This will stop transcription of the lactase genes. And again, this should make sense. We don't have lactose to break down, so why make lactase? Whenever there is high lactose present, now the cell needs to make lactase. So what happens is the lactose will bind to the repressor. This changes its shape. And again, this is another major theme of AP Biology. Shape determines function. So whenever you change the shape, you change the function. And thus, this repressor will unbind to the operator. When the repressor unbinds to the operator, this allows RNA polymerase to go through transcription and make the proteins of lactase. Again, now the cell has lactose to break down. Now it needs to make the lactase to break down those sugars. Now, whenever all of the lactose molecules are broken down by lactase, this lactose will be broken down and unbinds with the repressor. The repressor then will bind to the operator, stopping transcription. Now let's talk about a repressible operon. This is kind of the complete opposite. This is an operon that's usually on, however, it can be turned off whenever we have enough of a certain molecule. The example that most textbooks use is going to be the trip or the tryptophan operon. So again, in a repressible operon, it is usually activated where transcription and translation are occurring. However, whenever there's enough of a molecule, it's going to bind to the repressor, which is going to stop the transcription then. First of all, before we get into the trip operon, let's just remind you that trip or tryptophan is an amino acid. And within some prokaryotic cells, trip genes or tryptophan genes are used to make the amino acid of tryptophan. So again, we have our bacteria cell that has DNA, and there are going to be genes here, the trip genes, that make proteins that are going to make tryptophan. Whenever there is low amounts of tryptophan, the RNA polymerase is going to bind to the promoter, and it's going to transcribe these genes to make the proteins that make tryptophan, because again, the cell needs tryptophan. However, when there are high amounts of tryptophan, the cell doesn't need to keep on making tryptophan, it has enough. So what will happen is the tryptophan will bind to the repressor. The repressor will then bind to the operator. Once the repressor binds to the operator, it's going to stop transcription because again, the cell has enough tryptophan. Whenever the cell uses up the tryptophan, it's going to unbind then to the repressor. The repressor then will unbind to the operator, allowing RNA polymerase to transcribe the genes to make more tryptophan. It's important to remember that operon models or operons are going to be found in prokaryotic cells, not eukaryotic cells.